Good afternoon. You can hear me, guys? Please tell me on chat if you can hear me because it was not working the speaker. Okay. Okay, where's the others? Okay. So today we're gonna study bioenergetics. Today we're gonna study cellular respiration and photosynthesis. Yeah. So how does the cellular respiration happen in our body? As we were speaking about all this time, that the mitochondria is the reason that our cells will be respiring. This cellular respiration starts with a one glucose molecule ending up with so much ATP molecules from so many resources like NADH, FADH2, and other resources. So one of the first processes to start, let's draw the mitochondria. Let's have the outer membrane, the crista or the inner membrane, and it has the matrix inside. As we said, if there was oxygen, the Calvin, no. If there was oxygen, the citric acid cycle will continue, or the Krebs cycle. And if there was no oxygen, so here with O2, if there is no O2, no oxygen, we have fermentation will happen. Fermentation happens in the cytoplasm, not in the matrix. Inside the matrix or inside the inner membrane, the citric acid cycle happens and not the fermentation. So how is this? Let's first speak about the first process which happens for both the fermentation and the citric acid cycle. So both of them has a process which happens to create the pyruvate. From the pyruvate, if there was oxygen in this cell, it will continue and enters the matrix. If there was no oxygen, it will stay in the cytoplasm and undergo fermentation. So what happens here? We have first the number one process of making a glucose, a pyruvate, is glycolysis. Lysis means destruction of glucose. So okay, so first we start as we said, we start with one glucose molecule, and then it will undergo a reaction having an enzyme called hexokinase enzyme, making it glucose 6-phosphate. Usually kinases, one of the roles of kinases is to phosphorylate this molecule or to add a phosphate on this molecule. So it made the glucose to be glucose 6-phosphate. And then this glucose 6-phosphate, it will become fructose 6-phosphate, okay, by phosphofructinase. So phospho, this is the enzyme. Fructinase. This phosphofructinase enzyme, it helps in, by phosphorylation wait, biphospho. Here the enzyme is not phosphofructinase. Let's write it in this way. A moment, okay. Okay, so this glucose 6-phosphate will become fructose 6-phosphate by phosphoglucomutase. This one. So phospho. Hello, Saad. Glucomutase. So Saad, we were speaking about the glycolysis. We said that it happens in both the fermentation, which comes after this glycolysis, or the citric acid cycle. It depends. If we have oxygen, it will continue to have the citric acid cycle and then the electron transport chain. If there was no oxygen, it will undergo the fermentation in the cytoplasm. So we were saying that we start with one glucose molecule, which becomes glucose 6-phosphate. Why? Because the kinase enzyme, it phosphorylate or adds a phosphate for the molecule it acts on. And then this glucose 6-phosphate will become fructose 6-phosphate by phosphoglucomutase. What is the role of mutase here is for isomerism. So it helps the glucose to become a fructose. Glucose and fructose, as we said, both of them are hexoses. However, one of them is aldose and the other is ketose. This means that we have isomers. So it made the glucose to become a fructose. This is isomer, acting as an isomer, this enzyme, which is the mutase enzyme. And then this fructose 6-phosphate will become fructose. So we'll say fructose 1,6-biphosphate. These you are asked on, you should memorize them. So fructose 1,6-biphosphate by the phosphofructinase enzyme. So it already had a phosphate here. If you see, it already had one phosphate. We added another enzyme which contains a phosphor group or a phosphate group, and then it made one phosphate, and here we have the second phosphate. It added another phosphate, it became biphosphate, 2 phosphates. Okay, and then this fructose biphosphate will become 2G3P. Okay, or let's say 1G3P for now, and then I will say why they become 2. So it will become a G3P by an enzyme called adolase. Okay. So now this G3P, this fructose 1,6-biphosphate can become either 1G3P or it can become dihydrohyacetone phosphate. Okay, so it will give a 1G3P and dihydrohyacetone phosphate. Now this dihydrohyacetone phosphate, what will happen to it? It will undergo another reaction. An enzyme will act on it. Yeah, you have to memorize everything. Yeah, the pathway, the glycolysis, the enzymes, the why, what does the enzymes do, the names, everything. You have everything. Okay, so this dihydrohyacetone phosphate will undergo another reaction under an act of an enzyme, which is called triose phosphate isomerase. So first it will give, after the fructose 1,6-biphosphate, by adolase enzyme, it will give us 1G3P and dihydrohyacetone phosphate. Okay? Now this dihydrohyacetone phosphate will undergo another reaction using the triose phosphate isomerase enzyme to become another G3P. So what happens here? We have two G3P. So this is the final product of the first three steps of glycolysis. So here we already, no, not three steps, there are 5 steps. We already finished 5 steps of glycolysis. Or let's say 5 reactions. Let's not say steps because steps is something different. So we have here 5 reactions already done. Okay. And so they are usually 10 reactions. They are usually like 10 steps, 10 reactions. Now 5 of them are done. Okay. So for the glycolysis, we have 10 steps. And we have 3 phases. We will speak about the phases. Okay. And the delta G of glycolysis is, you should memorize it, it gives the free energy for glycolysis is minus 685 kilocal per mole. If you see, yeah, I will send Saad, don't worry. Okay, so if you see, guys, that we have minus 685 kilocal per mole. The delta G, which is gives free energy of glycolysis. So it is negative. If you see here, delta G is negative. This means that this glycolysis reaction happens spontaneously. Okay? And which also means that this reaction is exothermic. Why it's exothermic? Because it's emitting ATP or emitting energy. So that's why delta G is negative. Okay, minus 685 kilocal per mole. Sayed is 3 phases, not 2 phases. Perfect. Okay. So let me just number them. So from glucose to glucose 6-phosphate by the hexokinase enzyme, this is the first step. From glucose 6-phosphate to fructose 6-phosphate by the mutase enzyme, this is the second step. Hello, Angela. From the fructose 1,6, hello. From the fructose 6-phosphate to the fructose 1,6-biphosphate, this is the third step. Okay. And then from fructose 1,6-biphosphate to 1G3P is the fourth step. And then from fructose 1,6-biphosphate to dihydrohyacetone phosphate, this is the fifth step. However, from dihydrohyacetone phosphate to G3P, this is not considered a step. Okay. So, Angela, we were speaking about the glycolysis. We were saying that you have to memorize the enzymes, the act of the enzymes, why do they act, how do they act. You have to memorize the glucose, what does it become, the molecules, how many steps, each step, the phases, the delta G. And if you have any question, just tell me. I will try to explain it in a summarized way before we continue. So Angela, we start with one glucose molecule, and then it will become a glucose 6-phosphate by the hexokinase enzyme. The kinase enzymes usually phosphorylate or add the phosphate on the molecule they are acting on. And then the glucose 6-phosphate will become a fructose 6-phosphate. So if you see, if you notice that it was glucose, it became a fructose, which means that it was isomerized. So this means we use an isomerizing enzyme, which is the mutase. And then fructose 6-phosphate became fructose 1,6-biphosphate. Of course, if we have 2 phosphates, this means that we use an enzyme that contains a phosphate group. So we use the phosphofructinase enzyme. And then the fructose 1,6-biphosphate became a G3P by the aldolase enzyme. And then what happens? It also becomes a dihydrohyacetone phosphate, and then it undergoes another reaction by another enzyme, which is the triose phosphate isomerase, to become another G3P, ending up with a product of 2 G3P. Okay?
Okay, I understand it. Thank you.
Perfect. Okay. So now we spoke about the first 5 steps that we said. The 5 steps are the glucose to glucose phosphate to fructose phosphate, fructose phosphate to biphosphate, and then from biphosphate to G3P, and then from hydrohyacetone phosphate to G3P. Okay? So, now, number 1 phase, we have 3 phases, as we said. Number 1 phase is called the investment phase. So, or let's continue the steps and then we speak about the phases. So, first 5 steps, they are perfect. They are done. Let's go for the other steps and then we continue. So, we ended up, as we said, by 2G3P as a product, right? Now, we will start with this 6th step. So here what happens, since we have 2G3P, it will undergo, we will work on 1G3P. So we have 2G3P, let's work on one cycle and then we go to the other cycle, okay? So let's start with the first G3P. So 1G3P, it becomes 1,3 biphosphoglycerate. Okay, by the enzyme which is called dehydrogenase. So dehydrogenase enzyme would make this G3P into 1,3-biphosphoglycerate. The 7th step is 1,3-biphosphoglycerate, let's bring it here, will become 3-phosphoglycerate by a kinase enzyme. And then the 8th one is that this 3-phosphoglycerate will become a 2-phosphoglycerate by a mutase enzyme. And then this 2-phosphoglycerate will become 9. It will become PEP by enolase enzyme. And then this PEP, which is the last step, will become a pyruvate by kinase enzyme. Pyruvate usually have 3 carbons. So if you see that glucose, the C6 at 12O6 was 6 carbons, it ended up with 3 carbons after glycolysis. So glucose is getting decomposed, reaching the end of glycolysis. But we didn't finish yet. We didn't decompose it at all. We didn't finish from all the carbons. We only divided or decomposed only half of this glucose. Okay. So the enzyme here from PEP to pyruvate is kinase enzyme. So the other steps of making a G3P, a pyruvate of glycolysis. Now let me just, before I repeat, let me just say something. So here, as we said, that we ended up with 2G3P in the first 5 phases. So this is a cycle of 1G3P. If we want to see the whole cycle of this, glucose, you have to double it times 2. So 2G3P will be working here. If you want, let's say we write here 2 and we double all of this, okay? So it happens one time, this cycle happens 2 times, not just 1 time, because we have 2 G3. Okay. So ending up with how many pyruvate? Since we said we double it, it ends up with 2 pyruvates. Okay. This is the main product of the second steps of glycolysis. So now we have to repeat these phases again for you to try to memorize them. And then we have to speak energetically and we have to speak about ATP, how it goes inside, how it's invested, how it's used in the first steps, and then produced in the second steps. We will speak about all of this. So in the sixth step of glycolysis, G3P will become 1,3-biphosphogylycerate by the dehydrogenase enzyme. And then this 1,3-biphosphogylycerate will become a 3-phosphoglycerate by the kinase enzyme. And then this 3-phosphoglycerate will become a 2-phosphoglycerate by mutase enzyme. And then this 2-phosphoglycerate will become a PEP by the enolase enzyme. And then this PEP will become a pyruvate by the kinase enzyme. Only this process is repeated 2 times. Why? Is because we have 2 G3P ending up with 2 pyruvates. Okay. 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 Now we have to speak about. Let me try to write the steps in this way. Okay. So in the first step, guys, which is the glucose becomes glucose 6-phosphate. Okay. ATP will become ADP. As we said, it was adenosine triphosphate. It will become adenosine diphosphate. So 1 phosphate will be taken. ADP, usually we drew already this figure before, that ATP is the most energy from all of these kinds of phosphate adenosine. So tri-di-mono. So here we have using of ATP in the first step. In the second step, no. In the third step, yes. Also the same. So here we also have ATP becoming. So we already canceled 1 phosphate from this ATP. Why did we cancel? Because when we phosphate or when we add, when there is an enzyme that phosphates or phosphorylates the molecule, it will have to use a phosphate from this ATP. So they were like an ATP, we have phosphate, second, third. So it takes one of them and will give it to the glucose. So it was a glucose, it will become a glucose 6-phosphate. It added this phosphate, so here it becomes ADP. Same goes to the third one. It was fructose phosphate, it became fructose biphosphate. So also it had already one from the fructose phosphate from the reactant, and it used the ATP phosphate, which is the phosphate, 1 phosphate of this 3 phosphates that we have in the ATP, and it added them for it to become fructose biphosphate. So in the first one, in the third one, and then, so here we have energy change. So in the sixth step of the glycolysis, what happened? The ATP starts to be produced. So the most important step in the whole glycolysis is the sixth step. So is most important step in glycolysis. What happens in the sixth step? NAD plus will become NADH, okay, and it will produce outside H plus ion. And then in the seventh step, ADP will become ATP, so here ATP will start to be produced. And then in the ninth step, H2O will be produced, water. So these are energetically what happens in the steps. Okay, perfect. Now, so in the first step, we used this ATP. In the third step, we also use this ATP to phosphorylate the molecule. In the sixth one, until the end, we start creating ATP. So how many ATPs at the end of glycolysis? We're gonna have 2 ATPs. Why 2? Since if you see here, we only produce 1 ATP. Okay? If you see, like, if you try to look here, you will see there's only one, but we said that from 6 until down, this cycle will repeat 2 times because we are speaking about 1 G3P, not 2. If we were speaking about the 2 G3Ps, because usually one glucose molecule will produce 2 G3Ps, so this means that we will end up with also 2 ATPs because we said we will double the products. So glycolysis gives us 2 ATP, and it will give us 2 NADH will also be doubled, and it will give us 2 H2O. So, products of glycolysis, 2 ATP, 2 NADH, 2 H2O, and of course 2 pyruvate. Okay.
Sorry for the interruption, but I saw in a lesson that the product of glycolysis is 4 ATP.
No, it's 2.
It's 2 because they said glycolysis starts with one molecule of glucose and ends with 2 pyruvate.
Maybe. Yeah, I
Yes.
guess
No,
maybe
four
started
four are
with 2
produced, but
molecules.
2 are used also in preparative phase.
Yeah, exactly. Yeah.
She is right. Four are produced and 2 are used. That's why 2 are produced only.
Yeah, exactly. Yeah, exactly, 100%. Yeah, that's what I'm saying. I'm speaking about the end. I'm not speaking about all of glycolysis. I'm speaking about the end of glycolysis. What do we have? Not about the preparatory phase. The preparatory phase already used 2 ATP from them. So we don't have to speak about them or add them in the products. And in the IMAT, if they ask you what is the product of glycolysis, you have to say 2, not 4.
Okay, thank you.
So since we said in the first 3 phases we're gonna use 2 ATP, so just you have to take them out, minus. Anyways, so we spoke about glycolysis. Now, there's another thing. Yeah. So the roles of the enzymes that we use. We have the kinase enzyme. We have the adenylase enzyme.
One question, ma'am. How many
Yeah.
ATPs are produced during the whole aerobic respiration?
I'm
36,
not
right?
I don't understand. Can you please repeat? I didn't hear your question.
I said,
Who's speaking?
how many ATPs are produced during the whole aerobic respiration? 36, right?
Yeah, we will speak about the yield, don't worry.
Okay.
Perfect. Okay, so we have kinase, we have the adenylase, we have the isomerase. By the end of glycolysis, we will speak about the yield of everything products produced, so don't worry about it. And we have the mutase. Okay, so the kinase enzyme, so it's phosphating enzyme. The adenylase enzyme, it adds water. The isomerase mutase for isomerism. Okay, now we will speak about the three phases that we said at the beginning. So the first phase, which is phase 1 until 3, is called the investment phase. Okay, the second phase is from 4 until it's 4 and 5 only, I guess. Yeah, it's 4 and 5 only. So from 4 to 5, it's called the cleavage phase. And then the last phase is from 6 to 10. It's called the energy liberation phase. Investment phase is when we invest to ATPs in order to make this two pyruvates at the end. Okay. The cleavage phase is the phase where the two GTPs are produced, and the energy liberation phase when we produce already these ATPs. Okay. Okay. Now, one moment. Okay. What happens in the mitochondria during cellular respiration? So, glycolysis, glucose. So we have two steps. We have step 1, step 2. Step 1, we will have glycolysis. Means the glucose becomes pyruvate. Step 2, we have pyruvate oxidation to give us acetyl-CoA. So, after this glycolysis that happens, we are gonna have something called the pyruvate oxidation. This immediately happens after glycolysis. The pyruvate oxidation process is so easy. It's just a reaction you should memorize. So it starts with 2-pyruvate. It will give us acetyl-CoA. So which is looks like this. Plus 2CO2. Of course, here the NAD plus will be reduced. To NADH. And the enzyme which is used in this reaction is called the pyruvate dehydrogenase enzyme. So of course, since we started with 2-pyruvate, it will end up with 2-acetyl-CoA. Okay. So what else you should know? That this pyruvate oxidation happens in the matrix, and the glycolysis will be, so glycolysis happens in cytoplasm, citric acid cycle and or Krebs cycle in the matrix, pyruvate oxidation in the matrix, and fermentation in the cytoplasm or the cytosol. So okay, this is mainly the reaction of pyruvate oxidation. Now we're gonna speak about the oxygen and if we don't have oxygen. If we have a high amount of oxygen and this oxygen is enough for the Krebs cycle to start, aerobic respiration happens. If the amount of oxygen is not enough, okay, so it will make or it will undergo fermentation or anaerobic respiration. As we said, that this oxygen is gonna enter this is gonna enter the matrix by support the second active transport. So let's suggest that we have enough amount of oxygen, so we will undergo the aerobic respiration or the citric or Krebs cycle. So citric acid cycle. We start, of course, with citrate. Here also you should memorize everything. Okay. We have isocitrate, and then it will become alpha ketoglutarate. And then it will become succinyl-CoA. And then succinate. And then fumarate. And then malate. And then oxaloacetate. And then citrate again. And this the cycle continues. Okay. Before iso. No, it's okay. So you are saying about aconitate. It's not like something. It's not a kind of reaction that happens to become aconitate. We will use here an enzyme which is called the aconitase. But it's a very small reaction that happens, which is not this. With this detail, you should not know for the IMAT. It's not this deep for you to know for the IMAT. So you should just know that this enzyme will make the aconitate, and then this aconitate will become isocitrate. You should not know this. But you should just know that the enzyme that you're using here from the citrate to isocitrate is aconitase. Because for the IMAT, like sometimes some teachers say that this is not important. Even these names are not important. But I guess, like for me myself, I went to memorize. Like I memorized them and I go, and I went okay. So here the enzymes are from citrate to isocitrate. We're gonna use the aconitase enzyme. From isocitrate to alpha ketoglutarate, we're gonna use the isocitrate dehydrogenase. It's so easy, the enzymes, because you just take the name and you add the dehydrogenase for them. Okay, so isocitrate dehydrogenase. From alpha ketoglutarate to succinyl-CoA, we will use the alpha ketoglutarate dehydrogenase. So alpha keto dehydrogenase. From succinyl-CoA to succinate, we're gonna use succinyl-CoA synthase. From succinate to succinate to fumarate, we're gonna use succinate dehydrogenase. So also succinate. And then from fumarate to malate, we will use fumarase. And from malate to oxaloacetate, we will use malate dehydrogenase. So just like you write the name, you add the dehydrogenase. This is the enzyme. Because so much dehydrogenase enzymes are used in this cycle. And then from oxaloacetate to citrate, we will use the citrate synthase. Okay. Of course, we're gonna have 2-acetyl-CoA, or we will start with 2-acetyl-CoA before even the citrate starts. We use this acetyl-CoAs to create the cycle. However, we have 2-acetyl-CoAs means that this cycle is multiplied by 2 happens 2 times. Okay, because we have 2-acetyl-CoA. Okay, perfect. Now we will speak energetically about the ATP and these things. So let us name this cycle into 1, 2, 3. From here to here 1, from here to here 2, from here to here 3. Then from here to here 4, from here to here 5, 6, 7, 8. Okay, let's name them 8 steps. So before we continue, the isocitrate is 6 carbons. The alpha ketoglutarate is 5 carbons. The succinyl-CoA is 4 carbons. The succinate is 4 carbons. The fumarate is 4 carbons. And the malate is 4 carbons. And the oxaloacetate is also 4 carbons. Okay, what else you should know for the IMAT? Okay, so let me try to draw. Okay, so in the first step, we will use NAD plus to produce NADH plus H plus. Okay, and of course we will also produce CO2. And then here we will use from alpha ketoglutarate to succinyl-CoA NAD plus to produce NADH plus H plus and another CO2. And then from succinate, which is here, to fumarate, one FAD plus will become FADH2. And then from malate to oxaloacetate, NAD plus, it will be also NADH plus H plus. Okay, other things you should know that one ATP can be produced in the cis-alpha CoA here. It can be only one ATP can be produced. Why? Because so first it will use a GTP, which will make a which will decompose to make a GDP plus one inorganic phosphate. And because of this cycle that happened here, an ATP uncontrollably will be produced. Why? Because this GTP is decomposed. So this ATP will be produced from ADP. So we're gonna have one ATP produced. Okay. So what are the products of this citric acid cycle? They will be one CO2. One CO2 means two CO2. Okay. They will be one NADH, one NADH, and one NADH, three NADH. Okay, we have one FADH2 and we have one ATP, which is here. And what else? Yeah. So two CO2, three NADH, one FADH2, one ATP. Now, since we said that this cycle is multiplied by 2, so we're gonna have 4 CO2, 6 NADH, 2 FADH2, and 2 ATP. Clear? And also 2H2. It will be the water will be produced also during this cycle. So, Saad, what did you say? Sandy cycle with enzymes names how many ATPs are used to. When you can, I send a picture. Yeah, of course you can if you want, Angela. If you have picture, yeah, you can of course send on the group. I won't make a problem. Don't worry. Send the cycle with. I will send, and Angela will send. I'll try to send my notes because I have these as notes, and of course, like if you want, I can send you. When you can send, yeah. How many ATPs are used to? No, here not ATP is used. Here ATP is produced. No ATP is used here. We are producing. There is no investment here. We're not using no ATPs. Okay, Angela sent the picture. Let me see it. Yeah, exactly. Thank you so much, Angela. I will send another one. One moment. Let me send a second. So, study. Okay. Okay, guys, I will send you another one. Let me just make sure it has all the enzymes. Yeah, perfect. Yeah. This citric acid, where's the glycolysis? Yeah. Um, You check the group, guys. Okay. Did you see it, guys? Did everyone check the group? You saw it? Perfect. See, I told you to study from here, guys. This notes here, everything is so clear and so summarized. So try to study this from here. Even the evolution is so clear inside it.
This
Yeah,
notes
it's
is not vis
inside.
this notes is not accessible for me.
You mean like you want more enzyme, like you want more.
No, I was edit after
You're not able to open them.
yeah.
Really? How's this?
I was edit after the after 2 3 days of classes after the classes started.
Okay. I will open them for you. Okay,
Yeah.
I will send them. In.
Resend it, please.
Everything is not everything or just this PDF. Is it all the PDFs?
No, this IMAT notes. I don't have any notes from this IMAT alpha.
Okay, try to open now. Let me see. Yeah, perfect. You saw them. Okay. What are you talking about? No mnemonics. You mean like sentences to memorize? No, I never like tried this way. You know how I memorized them? I was like, okay, like kinase phosphate, like as the phosphate, I add a phosphate. This one make it isomer. I do it, like in this way. Like I memorize it in a way, understanding, Now, in fact, like if you see, let me do something to try to make you memorize it. 1 minute. Okay. Glucose, it will stay glucose, but it will add a 6-phosphate. And always memorize 6. Imagine glucose has 6 carbons. Always memorize we're gonna have a 6 in the product, okay? Glucose 6-phosphate. Glucose 6-phosphate, it will become, if it, take this glucose off and make it fructose. Fructose 6-phosphate, everything else will stay the same. Fructose 6-phosphate, you just add 1. Fructose 1,6-biphosphate, 2 phosphates now. Fructose 1,6-biphosphate, G3P. And the other one, dihydroacetone phosphate, don't memorize it. You won't be asked about it. And then G3P, it will be 1,3-biphosphoglycerate. This you should memorize it. 1,3-biphosphoglycerate. And then this 1,3-biphosphoglycerate, take this one off. 3-phosphoglycerate. This 3-phosphoglycerates, make it -1, which the one that you took it off, it will be 2-phosphoglycerate. And then 2-phosphoglycerate, PEP, and then PEP pyruvate. It's not really hard to memorize, but it's just somehow complex. But the names are like, you just try to make them in your mind and memorize them, But like mnemonics, it's really like hard to find for this. It's so hard, The enzymes, you don't have to memorize the names of the enzymes. They're not really hard also. The enzymes, you should just memorize each one, what does it do, yeah. you start kinase, mutase, phosphofructinase, and then aldolase, and then dehydrogenase, kinase, mutase, the same like cycle. Start with number 1 kinase. You don't have to write the names of the enzyme, you just have to memorize, is it a kinase, is it a mutase, is it a isomerase, you know, and just these are asked for the IMAT. So remember this cycle: kinase, mutase, fructinase, aldolase. This is the first 5 steps. For the other steps, you have dehydrogenase, and then kinase, same cycle again, mutase. Instead of aldolase, you will have enolase, and again kinase. This from 6 to 10, this from 1 to 5. Kinase, mutase, fructinase, aldolase, dehydrogenase, kinase, mutase, enolase, kinase. And if you see, so much kinase is used. However, in the citric acid cycle, you should not memorize no enzyme. The only enzyme that you should memorize is that the dehydrogenase is used. The dehydrogenase. Just memorize dehydrogenase is used in the citric acid cycle, nothing else. Because it's the same dehydrogenase, but changing the name. We are using a kind of dehydrogenase for each single cycle. Like to go from alpha ketoglutarate to succinyl-CoA, we are using alpha ketoglutarate dehydrogenase. To go from fumarate from succinate to fumarate, we are using succinate dehydrogenase. So it's a kind of dehydrogenase at the end. So we're gonna use only dehydrogenases and synthases in the citric acid cycle. However, in the glycolysis, we have kinases, mutases, enolases, aldolases, dehydrogenases, fructinases. It's not really hard, but you have to try to practice and memorize them. Okay, so we spoke about the products. Okay, perfect. So what did we say? We already said that the cycle will be repeated twice, and we spoke about the products. After the cycle, we will have an ADH, we will have FADH2, and these NADH and FADH2 will need to do something, because at the end of all of this aerobic
Ma'am,
respiration,
do you have glycolysis cycle with enzyme names? You sent it, but the enzymes name are not mentioned.
the enzymes are not mentioned. The
Yes.
glycolysis.
Yeah. You send it, but the enzymes.
Okay, let me try to find something. One moment. Give me 1 second. I'll send it to you. Okay, did you check? Perfect, Sad. Okay. طيب. So after the citric acid cycle, we're gonna have an ADH, we're gonna have a FADH2, but we want to reach at the end ATP. We want to reach at the end energy we want to reach at the end an energy source, you know. So what happens here? They will go all of them to the mitochondrial membrane, okay? And what happens in this mitochondrial membrane? The oxidative phosphorylation and electron transport will happen. So what happens? They will be oxidized, this NADH, this FADH2, using the H plus electron gradient and using ATP synthase to be able to produce the ATP. So how this happens? Let me explain it in a better way. So let's say here we have the cytosol of the mitochondria. Here we have, no, let's do this. Let's say we have here the outer membrane. Here we have the cytosol. Then here we have the inner membrane. And on this membrane, we will have complexes. Yeah. So what happens here? Here we have complex 1. Here we have complex 2. Here we have complex 3. Here we have complex 4. Here we have the laser unit, which is the stalk. So what happens here? We're gonna have here H plus ions, okay? So much H plus ions up. So how does this electron, so this is the ETC or the electron transport chain. How does electron start? Okay, so let's say we start, we said that we end up with 3 NADH and 2 and 1 FADH2. I'm speaking about only one cycle, not the 2 cycles together, because usually it happens on 2 cycles. So this 3 NADH, the electron will start moving from a complex to another, reaching the basal unit, to be able to produce the ATP, or to be able to transform this NADH to an ATP. So what happens here? Electron will move, reaching the first complex, and then we'll go to the second complex, and then we'll go up, third complex. Here the electron reached the upper membrane, I mean the inner membrane, which is up, and then here it will go back to the complex 4, and at the end it will end up in the basal unit. This is the chain of this electron transported from a complex to another. Okay, here we have head piece. Okay, what happens here when this electron is transferring or transforming from one complex to another? So this hydrogen which are outside, they will start to come here, which are in the cytosol, will start to come inside this gradient, this protons, producing water, H2O, making this reaction. The reaction of these two is 2 electrons plus 2 protons plus half O2. Okay, and it will give this H2O. And if you see in the IMAT, what will they ask you? The main question of all of this complex in front of you, it will only be what is the final electron acceptor in ETC. It will be oxygen. Oxygen, not oxygen ion, oxygen O2. So the only question in the IMAT that is asked about electron transport chain, what is the final electron acceptor, which is oxygen, this one. From this reaction, it's the final electron acceptor. However, the NADH and the FADH2, what happens to them? NADH will become NAD plus after this electron transport chain by producing one H plus to the outside, okay, and FADH2 and getting one to the inside. And FADH2 will become FAD plus by also doing the same process, ending up with creating ATP because of this transformation. Okay, so here we have, so the thing that you should also memorize about this chain is 1 NADH is gonna give 2.5 ATP and 1 FADH2 will give 1.5 ATP. Okay, so then how is ATP produced? If you remember, we drew here the basal unit. This basal unit will have the stator on the top and will have the head piece on the bottom. So what happens here in this basal unit when all of this transport of electrons happens, when these protons are transferred from a place to a place, what happens here? Here the reaction ADP plus N, it will take from outside an inorganic phosphate to create ATP, and this process will happen to create ATP from NADH from FADH2. It will also be creating water additional to that. Okay, and don't forget, oxygen is the final electron acceptor. Should we learn 2.5, 1.5, or 3? No, 2.5, 1.5, not 3. 3.2 is old one. 2.5, 1.5 is the updated one. Okay. So this is about the electron transport chain and the oxidative phosphorylation. Okay, so is it clear for everyone? I want to ask a question. Who is the one on the group named Sartak Shan? Is he with us here? Is it you, Rafi? Rafi. Ah, your name is Rafi, means friend in Arabic. Rafi Rahman. Okay, this Sartak Shan, he never enters with us a meeting. Is he with us or like he's from outside the group? I'm not understanding. Hmm. Yeah, maybe. I don't know. Because he always reacts on the group, but I never see a reaction on the meeting, so it's kind of weird. Okay. Now, We're gonna speak if we don't have oxygen, what will happen. So, we don't have oxygen, anaerobic respiration, which is the fermentation. We have two types of
Ma'am,
fermentation.
I want to ask
Yeah.
one thing. Yesterday,
Yeah.
did you teach glycogenesis and glucogenesis?
No, not yet. We didn't
Okay,
reach
okay.
gluconeogenesis yet. Don't worry,
But
we will reach.
oh,
So, now we will speak about the fermentation. There are two kinds of fermentation. One fermentation which is for yeast, which is the alcohol fermentation, and the other fermentation which is for normally animals. So we have two anaerobic respirations. We have the lactate respiration fermentation. And we have the alcohol or the yeast fermentation. So as we said, usually what happens, guys, we said that usually in the cytoplasm, glycolysis happens. If there is oxygen or no, glycolysis will happen. So we will start with the glucose molecule in both of them, normally. And then here we have glycolysis. Here we also have glycolysis. After this glycolysis, of course, we will produce ATP from ADP and inorganic phosphate. And then we will end up with 2-pyruvate as usual. And here we also will end up with 2-pyruvate. Now we reached this stage and we didn't find enough oxygen. What will we do? It will undergo fermentation. For the lactate fermentation, this 2-pyruvate looks like this. As we said, it's a 3 carbons, so it looks like this. Okay, and then this 2-pyruvate will produce lactate, which looks like this. Okay, of course, using NADH plus 1 proton from the glycolysis. So from glycolysis, use NADH plus 1 proton, producing NAD plus as substrate. So here, this is lactate. So what are the products of lactate fermentation? There are 2 NAD plus. So of course here the cycle repeats 2 times because we have 2-pyruvate. So here we have 2 and 2 for everything. So 2 lactate, 2 ATP, and 2 NAD plus. So products, we have 2 lactate. Okay. We have 2 ATP, 2 NAD plus. However, the alcohol fermentation, which is the yeast fermentation, okay, so it starts, we end up in 2-pyruvate as we said, also like it
ma'am,
makes the same
should we
thing.
memorize this cycle?
Yeah.
We just memorized lactic acid fermentation occur in humans and alcoholic occur in yeast.
You should memorize this cycle also. Nothing, it's not really hard. Glucose become undergo glycolysis. Glycolysis becomes 2-pyruvate. Pyruvate will become lactate from the name. Here the same. Glucose undergo glycolysis. Glycolysis will make 2-pyruvate. 2-pyruvate will make acetaldehyde. Acetaldehyde will make alcohol. We end up with the same, with the name. It's so easy. It's not really hard. Here, it's so easy. From pyruvate to lactate. Here, it's somehow just one additional step, which is the acetaldehyde. So what happens here? Glucose, we have, it undergoes glycolysis. It will make 2-pyruvate. This 2-pyruvate will produce 2-acetaldehyde. But you should not memorize the shape, I guess, only. And then this acetaldehyde will become alcohol or ethanol, let's say. Makes more sense in animals. So the product are 2 NAD plus, normally like the lactate fermentation, 2 ATP like the lactate fermentation, 2 ethanol, and of course 2 CO2. Why 2 CO2? Because this, when the 2 acetaldehyde become alcohol, it will produce CO2. So here it produces the CO2 in this reaction. Okay, and perfect. Another note you should take is every, let me write it again. No, wait. Yeah, 2 ATP in the first cycle, 2 ATP in the second cycle. 32 ATP are formed by chemiosmosis, lactic and alcoholic. Exactly. Okay, perfect. Okay. So, the note that you should take, as we said, so every glucose molecule changes 10 NAD plus into 1 NADH. Another note you should take is electron transport chain find electron acceptor is oxygen. Another note for a classic fermentation, the electron acceptor must be organic. Must be organic. Okay. Perfect. Okay. Another question?
Ma'am,
Yeah. Yeah.
how many ATPs are produced? One from lactic acid and one from alcoholic, right?
No, there are 2 in the lactate fermentation and there are 2 in the alcohol fermentation. Now there are 1, usually there are 1, but we have 2 pyruvate, so the cycle repeats 2 times. That's why we say 2. But there are 1 if the cycle is only 1, you know? If you speak
Okay, so
about 1 pyruvate, it's 1 ATP. If you speak about the 2 pyruvates that are produced from 1 glucose molecule, it's 2. 2 for
means
alcohol,
four
2 for
four from anaerobic respiration, 4 ATPs
4 ATP from anaerobic.
respiration,
No,
right?
no. Anaerobic respiration of alcohol, it's so different than the lactate. It's 2 from, not like you cannot add them together, you know, like alcohol is something and the lactate is something. So it's 2 from anaerobic for each one of them, you can say. But you cannot add them together, you know? Okay. What I also want to say, Yeah. Does the yeast have mitochondria? Does the yeast have mitochondria? Bravo. Bravo, Sayed. Okay, so since it has mitochondria, why does it undergo alcohol fermentation? Why doesn't it undergo usual aerobic respiration? Since it has the ability to undergo both of them. Bravo. So it's usually yeast usually can undergo both of them, can undergo aerobic and anaerobic, both of them. However, it prefers to undergo the alcohol fermentation because it's the most preferable kind of respiration for yeast. So for you, you should know that yeast have mitochondria. Another thing, does the mitochondria has can mitochondria undergo active and passive transports together? Can mitochondria exactly? How? Can someone explain this? How? So mitochondria is usually a normal cell in our body. As a liver cell wants to respire and get oxygen from the lungs, mitochondria also wants to respire and get oxygen from the lungs, right? So there is the fusion of oxygen and CO2 between mitochondria and the pulmonary and the arteries between the lungs and the veins between the lungs and the mitochondria. However, so there is a diffusion, means passive transport. However, when it uses this oxygen, Angela, the question is, does the mitochondria undergo both of them, passive and active transport? The answer is yes, because mitochondrial cells usually need to respire as any normal cell in the body. They really need to respire as the liver cell, brain cell, heart cell, whatever. So what happens here? This mitochondrial cell will be able to respire oxygen by diffusion normally, and will be able to use this oxygen that it respires it can be used to undergo the aerobic respiration using this exact oxygen. And to undergo the aerobic respiration, you will have the active transport by symport. So it uses the passive transport to apply the active transport. This is how mitochondria works. Another thing, does mitochondria produce water? Bravo. And does mitochondria have linear DNA? No, it has only circular DNA. Perfect. Now, after we finish the fermentation and the glycolysis, we will do one last thing before we end the session. Today's session was really only about bioenergetics. So, today I want to make it bigger, speak about photosynthesis and these things. How do you create it? We will just be speaking about the yield. Now, you were speaking before we start speaking about the yield, Saad and Sayed. You are saying that 32 and 38. Can anyone like explain for me or open mic and explain for me how did you reach 32 and 38? And
No,
say
I
you
said
said that, yeah, 10.
32 ATPs are produced by phosphorylation, from chemiosmosis, and 4 are
So you
produced
mean
from
you mean you speaking about aerobic respiration?
yeah.
Okay, 32. And Sayed, what did you say? How many?
He was saying
He said
total,
for
total.
ideal. He said for ideal respiration, it's 38. He said this. Ideal 38. Okay. How did you get 38? Or you said, how did you get 32? How did you do this? Can I know what did you add together?
No, I said 36 because two of them are supposed to be used in glycolysis, and
Mm-hmm.
32 from chemiosmosis and 2 from
Okay.
other processes.
Mm-hmm. Okay, now let's do the yield. So you guys, you already went through this topic, I guess. So you wanted to speak about the yield so fast. Let's go for the yield. Yeah. So cellular respiration yield. Okay. We start from a glucose molecule, undergo glycolysis. Okay, and then produces to pyruvate. Then this to pyruvate will undergo pyruvate oxidation. And then pyruvate oxidation will produce to acetyl-CoA. Then to undergo citric acid cycle. Okay. So glycolysis will produce, so on the substrate level phosphorylation to ATP, as we said, and we said it will produce to ATP and to an ADH. To pyruvate and to H2O. This glycolysis. Now, and then to the pyruvate oxidation here will produce two acetyl-CoA, which is here, and will produce two NADH, and it will produce two CO2. And for the citric acid cycle, we said it produces 6 NADH. I'll speak about the both cycles together. 2 FADH2, 4 CO2, 2 H2O, 2 ATP from the GTP, the one at the succinate, if you remember. So at the end, we have how many H2O? We have here 2 H2O, we have here H2O, we have 4 H2O as a total from all of this. How many CO2? We have 2 CO2 here, we have 4 CO2 here, we have 6 CO2 as a total. How many ATP? We have 2 ATP, NADH is 1.5. So this means it's 2.5. So this means here we have 5 ATP. Here we have 2 times 2.5 is also 5 P. Here we have 15 ATP. FADH2 is 1.5, means we have here 3 ATP. And if we add them, we will end up with 32 ATP as a yield. So 6 CO2, 4 H2O, and 32 ATP. Usually CO2 is not produced in the lactate fermentation, it's usually produced only in the alcohol fermentation. Another note you should take for this topic is the mitochondrial membrane is leaky to H+. Other thing about this, let me see if I have any other notes. Okay. This is mainly for the IMAT. 2H+, the proton.
There is confusion.
A lot of positive answers are there if you also consider the mitochondrial shutdown. Can you repeat? What should I repeat, Said? Yeah, tell me, Saad. Yes, Saad.
I'm confused. In my book, there is written there are total 36 ATPs are produced during the whole aerobic respiration, any cellular respiration, but you wrote 32.
Okay, let me try to add them. Yeah, if you add them, there are 32. Um, 36, I guess they took that one NADH is 3 ATPs and FADH2 is 2 ATPs. So since they took they took into consideration each one is this, and then they said that the 2 ATPs which are produced. Okay, let me tell you how your book is working.
I
So
guess
your book,
they took 2 from glycolysis and 2 from Krebs cycle, and remaining
no,
32 from electron transport
no,
chain.
no, I tell you what they did.
Okay.
They took that one NADH is 3 ATP, and they took one FADH2 is 2 ATP. And if you remember, I told you that one GDP will be used to create ATP in the citric acid cycle. They didn't take this into consideration because no book take it into consideration. The ATP which is produced from the succinyl CoA or during this reaction. So they don't take it into consideration. I guess we should take it into consideration, but they didn't take it into consideration. So 2 ATP, they took them out. The one of citric acid cycle, they said that citric acid cycle does not produce ATP, they just produce NADH and FADH2. So take 2, do this calculation, you will end up with 36. Another thing.
So total 36 ATPs, right? Total 36, right?
No, it's not 36. It used to be 36 from like 2
Yeah.
years. It used to be 36 from like 2 years.
I think my book is old version. Exactly, you're right.
Is an old book. Yeah, the updated version for the IMAT says it's 32. It's not 36. They just
Okay.
a kind of no, Said, it's not 38, it's not 36. Take it as 32. I asked someone who knows here in Italy, they said it's 32. So take 32 into consideration. The amount of ATP doesn't have a fixed value. It depends on wait, Rafi. The amount of ATP doesn't have a fixed value. It depends on what you take for each value. Yeah, exactly, 100%. It's not, it does have a fixed value, but each book writes something, so they make you like, what's this, you know? But like you just should take the one which is of the updated version for the IMAT. Okay. So now it's 6. Okay. So now it's 6, 6. I guess we'll finish the session now. Okay, that's what I mean. Ma'am, 6H2O are produced, right? Yeah, 6H2O, exactly. Okay. So I'm gonna end the session now. One second before I end the session. Yeah, I wrote 4H2O. Yeah, exactly. Yeah, I read what you said, sorry. Yeah, it's 2H2O from glycolysis. And it's 4H2O from citric acid cycle. Because I forgot to multiply by 2 the water, you know? So it's 6H2O, 6CO2, and 32 ATP. Okay, perfect. So I will end the session now. Tomorrow, we will speak about gluconeogenesis. I'm checking the syllabus and seeing if we still have anything which is hard, like a topic which is hard. So I was seeing that it's someone is taking me out of the meeting. What's going on? Can you hear me, guys? Okay, what happened? Why I was out? Okay. Oh my God. Okay, so I was seeing the syllabus of the bio. I guess for the bio, we still have the systems of the body. Okay, we did all of these. Yesterday we spoke about the redox reactions. Guys, try to, who didn't study about yesterday we did 4 hours, mostly chemistry. So who was asking, please focus on chemistry. Yesterday I focused so much on chemistry. Okay, we have to study about tissues, we have to study about epithelium, we have to study about
Genetics.
genetics, exactly. Yeah, Mendelian genetics.
Because it's
And enzymes, the roles of enzymes. Okay, and the cells sizes. We studied about these. And what else? Yeah, genetics. Guys, I was seeing when I was speaking at the beginning with everyone, I found out that most of you has problems in genetics. Why is this? Is in Pakistan, because most of you are Pakistanis. So is in Pakistan genetics topic is not really explained well, or like it's something from your base? Okay, let me just
I'm